Right, class. So the nucleotides, as I already described these things in the class itself, nucleotides are the building blocks of nucleic acids. Nucleotides are the starting material of nucleic acids. Nucleotides are the monomers of nucleic acids. But what are nucleotides? Nucleotides themselves, according to these two scientists, are made up of three components: 5 carbon sugar. Levin and Alexander Todd in 1950 explained that they are consisting of three things, three factors, three constituents: a 5 carbon sugar, which is a pentose sugar, a nitrogenous base, and a phosphate group. Sugar, what sugar is this? This is actually a ribose sugar. If you see in RNA, the ribose sugar is present. Ribose sugar is simply appearing, as you know, most of the time about fructose, glucose, etc. So they are just like that. But if you look at DNA, in DNA it is deoxyribose. So obviously one oxygen is being removed. So why, at which location it is being removed? At 2' location. So you can see at this location, at this point, the oxygen has been removed. While here in this case there is no removal of such oxygen. I'm sorry, I have to show you like this. Yeah, look at this. So no removal of, it is a hydroxyl position. So this is ribose sugar, this is deoxyribose sugar. Fine. Now this is about sugar. Now second is nitrogenous bases. So nitrogenous bases, as I told you, they may be planar, they may be aromatic, they may be heterocyclic derivatives of nitrogen, or which are actually of two types: purine and pyrimidine. Okay, so purine is of two types, as I told you: arc cut, AgCut. So Ag is purine, and I told you to remember that purine are the small name, but their structure is big, that is double ringed heterocyclic rings, and pyrimidines are large name but small structure, that is just one ring. So adenine and guanine are double ringed structure, pyrimidine, and that pyrimidine is having cytosine and thymine, which is a single ringed structure. But in case of RNA, the thymine is replaced by uracil, which is also a pyrimidine. So do remember that all these 5 nitrogenous bases are well known to you. Focus on the structural differences. In case of thymine, there is a methyl group available over here. In case of uracil, in place of methyl, 2 oxygens are present, and that's why that's what I told you, the reason behind of it's not being present in the case of DNA. Fine. So do remember purine and pyrimidine. Yep. Fine. So now the association. Fine. So I have shown you out this in the class also, that at position number 1 this is adenine, and at position number 5 this is a phosphorus. So this is adenine, this is phosphorus, this is ribose sugar or deoxyribose sugar if you remove oxygen at position number 2. So this is right now called as deoxyadenosine 5-phosphate monophosphate, or you can write as DAMP, right? So do remember how you are going to name it. So nucleotide is phosphate ester monomer of pentose. Oligonucleotide is short polymer of less than 10 nucleotides. Polynucleotide is long polymer of more than 10 nucleotides. So nucleoside is the monomer of sugar and nitrogenous base only. Okay, so this I have described also. So this is the association. If you see at this location, this is the 5' end, and at this location this is the 3' end. So the 3' end makes an association with the 5' phosphate of the next incoming nucleotide. So this kind of bonding is actually called as phosphodiester linkages. This bonding is called as phosphodiester linkage, and the linkage between this nitrogenous base and the ribose sugar at this position is what we call it as this is the glycosidic bond. So this is phosphodiester linkage, and this is the N-glycosidic bond, where it is linking a nitrogenous base to C1 of pentose sugar. So this is, I have already told you that the polarity always goes from 5' to 3'. The first nucleotide having a free 5' phosphate, no bonding. The last nucleotide having a free hydroxyl end, with no actual, means no bonding. So these are the free components. So you are writing it from 5' to 3'. Okay. This is the actual representation. Now about features of DNA, as I have discussed in the class, I'll be describing in the next presentation. Thank you.